So welcome to Shankar Medico Academy. My name is Gnana Priya, who is working as a faculty for Botany. And today's topic is about endomembrane system, which comes under the cell unit of life. So I have written the entire topic so that it makes you for the easy understanding of the content. So when you talk about an endomembrane system, it is a series of compartments that works together to package, label, and ship the proteins and molecules. So when you talk about a cell, so basically when we know what is a cell is a basic unit of life. So cell comprises of cytoplasm, that is we call them as protoplasm, where nucleus, cytoplasm, and plasma membrane is there. So this, which the entire structure gives a clear idea of what is a cell is. So inside the cell, there is a special structure called as endomembrane system. It is called a special structure called endomembrane system. So what is the basic function of the system is synthesize a protein, modify a protein, they pack the protein and label the protein, and they transport outside the cell. So when they transport outside the cell by a phenomenon called exocytosis. So what is endomembrane? It's a compartment where many organelles are present, and these organelles, what function they do? They synthesize, modify, pack, label the protein, and they expel out of the cell. We call this as exocytosis. So now we come for the next topic, or the next point under that, where it includes an organelle called nuclear envelope, endoplasmic reticulum, Golgi apparatus, lysosome, vacuoles. All these organelles are present in endomembrane system. So what was the first thing we saw? What is endomembrane system? Endomembrane system is a compartmental system where it is going to function or work together. What is endomembrane system? Where they're going to function or work together. What all the organelles are there? Nuclear envelope, endoplasmic reticulum. When you take it as an organelle, it is endoplasmic reticulum. It is only a part of a nucleus. So it is a part of a nucleus. What are the organelles are there? Endoplasmic, Golgi, lysosome, and vacuoles. All these are made up of single membrane. All these are what membrane organelle? It is a single membrane organelle. It is a single membrane organelle where the nuclear envelope is a part of a nucleus. So to come from first, what is an endomembrane system? It's a compartment where it is going to work together, and they're going to pack and they're going to ship the protein. So what all the organelles are included? Endoplasmic, Golgi, lysosomes, vacuole. All these organelles are single membrane. What does they do? They synthesize, modify, pack, label, and finally what they do? They do exocytosis. What do you mean by exocytosis? They transport outside the cell. So this is what is about your endomembrane system. So now we come for the next. We'll see the pictorial representation of how does the endomembrane system occurs. So this is a cell. I have drawn a cell here, and this part is called as a nucleus. This, the entire part, we call it as what? Nucleus. This is endoplasmic reticulum. Since it is studded with red color thing, we call that as ribosomes. We call this endoplasmic reticulum as rough endoplasmic reticulum, and the green color one we call it as Golgi apparatus. The pink color which is represented is the plasma membrane of the cell. So I have drawn in this fashion. So nucleus, rough endoplasmic reticulum, Golgi apparatus, and plasma membrane. You understood now? Fine. This is nucleus, rough endoplasmic reticulum, Golgi apparatus, followed by plasma membrane. So now we come to the function. So basically, this is a nucleus. This is a nucleus, and this yellow color region we call it as nucleolus. This yellow color region we call it as what? Nucleolus. This yellow color region we call it as nucleolus. The orange is we call it as chromatin reticulum. So this is a typical structure of the nucleus. This is a typical structure of a nucleus. Rough endoplasmic reticulum, Golgi, and plasma membrane. Inside the nucleus, we have a yellow color region. The stained region. This stained region we call it as nucleolus. The stained region we call it as what? Nucleolus. It is rich in RNA. This nucleolus is rich in what? This nucleolus is rich in RNA, and they produce what? mRNA. They produce what? mRNA. I hope you all know a central dogma theory. According to that, what? DNA is converted to mRNA. mRNA is converted to protein. So DNA to mRNA, we call the concept as transcription, and this we call it as translation. So now what happens? From the nucleolus, understand clearly, from the nucleolus, the mRNA is getting transferred to rough endoplasmic reticulum. Since the mRNA moves to the rough endoplasmic reticulum, it will go and attach to all the mRNA will go and attach to the ribosome. So what happens? The rough endoplasmic reticulum gives the surface for the attachment of the RNA. So whatever the RNA coming from the nucleolus will come and attach to the ribosome present in the rough endoplasmic reticulum. Now what happens? What does the mRNA is there? Now what will be the next step? This mRNA will be converted. mRNA or what? They are nothing but the genes which code for proteins. Now what will happen? All the mRNA will be converted to amino acid. Amino acid will make a peptide chain. They become a protein. So what is the first step? From the nucleolus, the mRNA moves to the rough endoplasmic reticulum. What does the rough endoplasmic reticulum has got? Ribosome. Now it will go and make a protein. What we studied already, ribosome is a protein factory. Now the protein is made. Whatever the protein the rough endoplasmic reticulum make, it is an immatured protein. It is a what protein? Immatured protein. This immatured protein will be present inside the lumen of the endoplasmic reticulum. It will be present where? It will be present in the lumen of the endoplasmic reticulum. But what type of protein it will be? It will be in the form of an immatured form of protein. What protein it will be? Immatured protein. Now what happens? This immatured protein will come to the external region, that is to the external lumen of the rough endoplasmic reticulum. It is present inside, it will come out and it will transport to the Golgi apparatus. This immatured protein will go to the Golgi apparatus through a small vesicles. Through small vesicles, it will move to the Golgi apparatus. Now, what happens the Golgi apparatus? What is the function of the Golgi apparatus? They modify, pack, label the protein. What the function they do? They modify, pack and label. MPL function they do. What does the Golgi apparatus do? They modify the protein, they pack the protein and they label the protein. So the immature protein will come to the cis phase. This part we call it as cis phase. When the Golgi apparatus facing the nucleus, when the Golgi apparatus facing the nucleus, we call it as cis phase. When the Golgi apparatus, the part of the Golgi apparatus facing the plasma membrane is called trans phase. Now what happens? Once again, I'm coming from first. From the nucleolus, who is moving? RNA. Move to where? RNA moves to the rough endoplasmic reticulum. This RNA will go and bind to the ribosome and they become a protein. This protein is what form of protein? Immature protein. This immature protein will be packed in a small vesicle and that vesicle will move to the Golgi apparatus. Now, which part of the Golgi apparatus? In the cis part of the Golgi apparatus. Once it went to the cis part of the Golgi apparatus, what will happen there in Golgi apparatus? They get modified, packed and labeled. Once modified, packing, labeling is done, the vesicles will move to the trans phase. The trans phase, what will happen? They become a larger vesicle because of that modification and packing and folding of the protein. They become a large vesicle. This large vesicles will come out of the Golgi apparatus and they fuses with the plasma membrane. They come out of the apparatus, fuses with the plasma membrane. When it fuses with the plasma membrane, it will undergo a process called exocytosis. What is exocytosis? It will come out and it will make a vomiting. What do you mean by cell vomiting? Exocytosis occur. They will just expel out the protein. So this work, when it is done in a coordinated manner, we call the entire system as an endomembrane system. It is called as what? Endomembrane system. Do you understood? Once again, I'm repeating. Nucleus. From the nucleus, what does have? A densely stained structure called nucleolus. From here, the RNA moves via the nuclear pore, enters the rough endoplasmic reticulum. Here, the protein will be synthesized and folded. The protein is immature. Immature protein goes to the cis phase of the Golgi apparatus, where they undergo what? Modification, MPL. Modification, package and labeling, and they enter the trans phase. Once they enter the trans phase, the vesicle becomes bigger and go near the plasma membrane. So where do they grow? They go near the plasma membrane, and they fuses with the plasma membrane and they undergo exocytosis. What is exocytosis? It is called as cell vomiting. So whatever I have explained in the form of a diagram, I have made it in the form of a words to make a clear understanding of the concept of the same. So I have written a flow chart of what I said earlier. First, what is the first point? The nucleus, it is a source of what? RNA. It enters in the form of an mRNA via the nuclear pore. So the nucleolus lens, what is coming? RNA, and that RNA enters via the nuclear pore. Now it enters the rough endoplasmic reticulum. It attaches to the ribosomes. What are ribosomes? Protein synthesis. They are the protein factory. Now ribosomes builds the polypeptide chain. So third step is, first is RNA going to the rough endoplasmic reticulum. From the rough endoplasmic reticulum, ribosome is going to produce a protein chain. Now the chain is pushed to the lumen of the endoplasmic reticulum. Then it passes to the outside of the endoplasmic reticulum. They form what structure? They form a vesicle structure. They will form a what? A round structure. We call that as vesicles. What will be present in here? The proteins will be present here. So now the vesicle structure, which will be smaller size. Why? Because they carry a immature protein. Because they carry what? Immature protein, and that passes to the cis Golgi network. Now, once it enters the Golgi network, it undergo a modification, and a glycoprotein will form a matured protein. So they will form what? Once it enters the Golgi apparatus, they become a matured protein. Once they become a matured protein, they become a large sized vesicle, and they will enter the trans phase of the Golgi network. In cis phase, they will have what? Small vesicle. After the modification, they become large vesicle, and they enter the trans phase. Now what will happen? Once they enter the trans phase, the Golgi will fuses with the plasma membrane. Now what happens? Once they enter the gland, they will go and fuse with the plasma membrane, and they undergo exocytosis, which is expelling out of the cell. So what was the first point? First, it enters the nucleolus via RNA. RNA is going through nuclear pore to the rough endoplasmic reticulum, and where they have a ribosome, they build the protein. The protein which they build was immature, so they make a small vesicle. This small vesicle passes through the cis phase of the Golgi apparatus, where the cis phase of Golgi apparatus, they have a smaller vesicle which undergo modification, packing and labeling, and become a matured protein. This matured protein will become a larger vesicle. They go to the trans phase of the Golgi apparatus. Once they go to the trans phase, what will happen? They'll fuses with the plasma membrane, and they undergo exocytosis. By this process, or by this methodology, or by this way or a phenomenon, the endomembrane system, where it works with many organelles, and these organelles are single membrane, and the functions are done in a coordinated manner. What are the organelles involved in endomembrane system? Endoplasmic reticulum, Golgi apparatus, lysosomes, and vacuoles. So what is an endomembrane system? It is where it has got many organelles bound by a single membrane, and they do their function coordinatingly. So that was given in the next slide. I have written in the next slide of this topic. Endomembrane systems bound organelle, and they function in a coordinated manner. So where they function to be in a coordinated. So endomembrane system does not contain. What do they don't contain? They don't contain mitochondria, chloroplast, peroxisomes. They don't contain any microbodies. So microbodies like peroxisomes, glyoxisomes, none of these are present in the endomembrane system. So mitochondria or a chloroplast is not present in the endomembrane system. So what is basically an endomembrane system? It's many organelles are present. They are basically made up of single membrane, and they do a function in a coordinated manner, and they have compartments in it. So a compartmentalization where the functions are being coordinated by a different organelles to do the same, to do the single purpose. So that we call it as endomembrane system. So this is about the endomembrane system. So they show how from nucleolus rough endoplasmic reticulum, Golgi apparatus, and vesicles expel the protein out. So that's how a transport occurs between the cells. From one cell to another cell, when a transport occurs, we call this as a intracellular transport. So by an intracellular transport, the proteins are getting communicated outside the cell. So now you understood, I hope you understood clearly about the endomembrane system. We'll go to a question. For example, we'll solve the question based upon this concept. It's basically an assertion reason question. So based upon the concept, there was a question has been there and we'll solve this question. So once you understand the concept, definitely we can solve this question. So what was the assertion? The endomembrane system includes endoplasmic reticulum, Golgi complex, lysosomes, and vacuoles. Obviously, endomembrane system contains all this. So assertion is true. Exactly. What is the reason they have given? Mitochondria, chloroplasts, peroxisomes are not a part of a endomembrane system. True. This is also true. Reason is also seems to be true. And for till this part, it seems to be true. What is they given? What has been proceeded? Because their functions are coordinated with the same. Because their functions are coordinated with the same. So here the mitochondria also does the function in a coordinated manner. That's why they are not present in that endomembrane system. Here, both assertion and reasons are true, but R is not the correct explanation of the A. But R is not the correct explanation of the A. The endomembrane system includes endoplasmic reticulum, Golgi complex, lysosomes, and vacuoles. The reason is mitochondria, chloroplast, and peroxisomes are not present. That is true. The reason which they have given is true. But what was the explanation they are given? The explanation is because their functions are coordinated with the same. Not mitochondria, chloroplast, peroxisome. Their functions are not coordinated. Whose functions are coordinated? Endoplasmic reticulum, Golgi apparatus, lysosomes, and vacuoles. Only these functions are coordinated in manner. So because of their coordinated with the same. So the option is both A and R are true, but R is not the correct explanation of the A. Thank you. Thank you.